all three domains of life in the glycolysis pathway. They're believed to be very old enzymes and slowly evolving, which makes them really good candidates for this project and like looking at the beginning of life. So our hypothesis was that the more conserved sites at the root of the tree will be more likely to have the older amino acids.
So kind of the methodology that we used was first we do an alignment, and then we find a set of trees, and then we see if that's the right set of trees or the best set of trees, and then we look at our data. So when we're talking about an alignment of sequences, each row here represents a different species. So for ENO, we had 258 species that we had data for that enzyme for, and for ENO was 383, and FBPA was like 258 species. And so we have that many rows, and then each of them contains like variable lengths for the enzyme. Like some species the enzyme might be 350 amino acids long, one might be 360. And so we have to line them up because when species change and mutate over time, either amino acids can change, or they can be inserted, or they can be deleted. And so what we want to do is we want to take relatively okay, we think that according to this alignment, for all of these species, this is like the same site in their amino acid, like in this enzyme data. And so when we're talking about conservation, like a site like this one here would be a really conserved site because we see all of these are glutamic acid. But then a site like this one over here would probably not be very conserved because we see a lot of different amino acids at that site. And so that's what we're talking about with conservation.
Okay, so the software we used was Mr. Bayes, and so the model we used was the F81 model, which was created by Felsenstein in 1981. So the point of the model is to specify how amino acids can change from one amino acid to another one. And so this model allows that the frequencies of each amino acid to be different from the other ones. And so then what Mr. Bayes gives us as output, well, part of what it gives us is a vector of likelihoods for the 20 amino acids. So when we have our non-partition data sets, we have a vector for each site, which we're going to refer to as P, and then an overall vector, which we're going to refer to as pi. For the partition sets, we have a pi for each partition.
Yeah, and we'll talk about the partitions, right here. So when we're talking about partitions, what we're doing is we're like categorizing the data or like grouping it together. So the first thing we did was we have no partition, and so that's just like the sequence alignment as it is. We look at each individual site and we compare all the sites kind of together. Like at each site, we're going to compare the age and the conservation of that site, and we're going to plot them. When we're talking about the partition data sets, we use these two different scoring, the Shannon score and this conserve, and they calculate the conservation of the site a little bit differently, but basically they look at the observed amino acids at that site, and it says this is very conserved if they're all the same amino acid, or if it's not very conserved if they're all a bunch of different ones. And so when we have the partitions, we group them. So we have the most conserved, second most conserved, third most conserved, and the least conserved. So for both of these scores, we used 4 partitions to look at that. And we basically compare the partitions. We want the most conserved partition to reflect older amino acids and the least conserved partition to reflect newer amino acids.
Okay, so the algorithm that Mr. Bayes used